This set of notes will be on regulating gene expression. Remember, we talked about in the last set of notes protein synthesis and how that happens. We said that transcription is the first step, and in a eukaryotic cell, that step takes place inside of the nucleus. It starts with DNA, and then we create the messenger RNA strand from a gene of DNA. We also said that in eukaryotes, post-transcriptional modifications will occur. This will include a poly-A tail on the 3' end and a modified guanine cap on the 5' end, as well as the splicing out of the introns so that we only have coding sections leaving the nucleus and going to the ribosome, where the second step of protein synthesis, translation, will occur. We'll use the mRNA strand to create the polypeptide chain. A couple of vocabulary terms that you need to know. The genome is going to be the sum of all the genetic information inside of a cell. This will include both coding and non-coding regions. For humans, we contain approximately 25,000 different coding genes. And remember that your genome will be the same in all of your somatic cells. So remember, that means that the DNA inside of all your body cells, all your diploid cells, is identical. Same genetic code in all the cells. What's different is going to be your transcriptome and therefore your proteome. So the transcriptome is going to be all of the mRNAs that are being transcribed inside of a cell. So these are going to be the genes that are being expressed. Gene expression will be different from cell type to cell type. This not only varies between different types of cells of an organism, but gene expression and therefore your transcriptome and proteome will vary over the lifetime of that cell. So as a cell is going through changes, then this will also change. This transcriptome will also change. The transcriptome is going to be regulated. So we will talk today about ways that transcription will be regulated. The proteome is all the proteins that are being produced by a cell. And this, of course, is based on the transcriptome since mRNA is used to create the protein chain. How much of each protein you make and the types of proteins that you make is not only varying from cell to cell, but also through the lifetime of the organism, and this too will be regulated. So we'll talk about ways to regulate both transcription and translation today. The pattern of gene expression within a particular cell is going to determine what that cell is going to do, like what type of cell it is. This is called cell differentiation. Your cells, remember, will start off as unspecialized stem cells, but they will go through changes throughout development of the organism that will lead to specialized cells. This is called cell differentiation. And this is going to be controlled by controlling patterns of gene expression. Gene expression is highly regulated for multiple reasons. We need to be able to control what cells become. Remember, if they're starting off as a kind of generic stem cell, ultimately we need them to become specialized cells, skin cells, eye cells, liver cells, muscle cells. And this is really important to proper development of an organism. This is going to be controlled by turning certain genes on, turning certain genes off, and being able to regulate gene expression in a variety of ways. If all the DNA is identical inside of all of your cells, then why are there skin cells? Why are there liver cells? This is because certain genes are on in only certain types of cells and off in other types of cells. In addition to that, cells must make the right amount and the right proteins at a certain stage in development. And so an example is going to be that when you are a developing fetus, you make a particular type of hemoglobin that's called fetal hemoglobin. But as you grow into a child and then into an adult, the type of hemoglobin chains that you're making are actually different. So you'll then transition to make adult hemoglobin. And this is done by controlling gene expression. Genes are turned on, genes are turned off. In each cell of a multicellular organism, you would only express a small percentage of your genes. Expressing all of your genes would be chemical chaos, and it would not allow for cell differentiation to occur. So again, some genes are on and some genes are off, and this is going to vary from cell type to cell type. We're going to talk about ways that that is regulated in this set of notes. Gene expression is regulated by several different methods. It could involve proteins that bind to specific base sequences on DNA. So we are going to talk in just a moment about activator proteins binding to enhancer sequences, which will turn the gene on, thereby speeding up the entire rate of transcription. We're also going to talk about proteins that can slow down transcription. Those are called repressors, and they will bind to silencer regions on the DNA sequence. Please keep in mind that when transcription is sped up or slowed down, we're not saying that the process of transcribing each gene is occurring any faster. It's not like RNA polymerase all of a sudden gets to travel faster down the gene and make mRNA at a faster rate. It doesn't work like that. Transcription is either on or it's off. So when we say speed up the rate of transcription, you would need to consider like a whole tissue sample. If more cells of that tissue type have that gene turned on, then transcription is sped up, right? There's more transcription happening. The overall rate of transcription is higher. But if less genes have that particular, or less cells have that particular gene turned on and instead have it turned off, that would slow down the overall rate of transcription. Gene expression can also be controlled by environmental factors, epigenetics, the degradation of mRNA, so breaking down mRNA, and then alternative splicing. We'll talk about more of all of these on upcoming slides. We'll first talk about proteins that bind to specific base sequences on DNA in eukaryotic cells. So remember that only some of your DNA sequences actually code for proteins, but there are other important sequences of DNA that still provide a function. We talked about the promoter as one of these examples. The promoter is non-coding, but it still has a very important function. It's typically located near a gene, and its job is to bind RNA polymerase, the enzyme that performs transcription. So remember, the promoter is like a highlighter. Bind here, and then RNA polymerase binds at the promoter and can help to start transcription. Transcription will occur forming the phosphodiester bonds between the mRNA nucleotides. Transcription is actually quite a bit more complicated than that, and other proteins will also be involved. These other proteins are called transcription factors or transcriptional factors, and these transcription factors may be general, meaning they're not specific to the gene. They can be used by lots of different genes, or they can be much more specific to just a specific gene. And typically, an overall transcription complex may require both kinds. They may require general transcription factors and specific transcription factors. This first picture is pretty simple, just shows the promoter and the gene and RNA polymerase binding to the promoter. This one adds in some transcription factors, right? So they're going to also bind near the promoter along with RNA polymerase in order to help turn the gene on. Enhancers are going to be regulatory sequences on the DNA, which will increase the overall rate of transcription of a tissue type when activator proteins bind to them. So activator proteins bind to enhancers, and this helps to form this overall transcription complex, which I'll show you in a minute, and that's going to help to turn on transcription of that gene in that particular cell. Silencers are going to have the opposite effect. They are a region on DNA, a regulatory sequence on DNA, which binds a different type of protein called a repressor. When the repressor protein binds, it represses or turns off transcription. So we're either enhancing transcription or we are repressing transcription. We're turning it on, which speeds up the overall rate within a collection of cells, a tissue type, or we're turning it off, which slows down the overall rate. Activators and repressor proteins are examples of specific transcription factors that may be needed to turn transcription either on or off, regulating gene expression. Students often get confused with which term is part of the DNA and which term is the actual protein that binds to the DNA. So be really careful when studying these terms. You need to know that the enhancer is on the DNA, it's part of the DNA, and so is the silencer, versus the activator and repressors. They are proteins that bind to those regulatory DNA sequences. So I typically say to always say the term protein after the term activator or repressor. If you always call it an activator protein, instead of just calling it an activator, it might help you remember that that is the protein. So here's some pictures showing some of these molecules involved, and you will see that there may be other molecules involved too. When transcription is enhanced, it's being turned on. So RNA polymerase is going to bind near the promoter, and then there's going to be other molecules that come in and bind as well to turn on transcription. So we're going to have general transcription factors. Those are not shown in this image, but they are shown in this one, the green molecules. There may also be activator proteins. Those are the ones that bind to the enhancer. So that's this guy shown in green, or this guy shown in red. And then there may also be other necessary linking proteins. Like, for example, this one shows a mediator. Another term for a mediator is a co-activator. So you may hear it called both things. A mediator and a co-activator do the same thing. They link the activator protein to the rest of the transcription complex, so to our general transcription factors and RNA polymerase. So they kind of help bring everything together. I'm going to show you a short video that shows this complex on the next slide. So this gene has turned on. This loop, because we have the complex of proteins forming, helps to turn the gene on. The repressor does the opposite. It binds to the silencer region, and what it does is it blocks the activator from binding. So in this particular picture, here's RNA polymerase shown in this pink color. These green ones are the transcription factors, general transcription factors. These gold proteins are the mediators or co-activators. And then this is the activator itself. Normally, the activator would bind to one of these enhancer sequences, but when the repressor is bound to the silencer, it prevents that. So now we don't get the loop, we don't get the full transcription complex, this gene is off. So we're going to now watch an animation that will show both processes. It will show enhancing and silencing transcription.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TA-TA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
The video called these basal factor proteins, but you can just call them transcription factors. Okay, so here's another picture showing the full transcription initiation complex. in eukaryotic organisms, eukaryotic cells. So again, here's RNA polymerase at the promoter, general transcription factors in green, co-activators, which link the general transcription factors to the activators, helping to form the loop. And oops, going back. And the activators are bound to the enhancers. If there were silencers here, it would prevent the activators from binding using repressor proteins, but that's not shown on this diagram. This gene is on. So this is a way for cells to turn genes on or turn them off using regulatory proteins and regulatory sequences of DNA in eukaryotic organisms. This is a similar example, but a little bit different because now we're talking about regulation in prokaryotes using proteins. And so we're going to be talking about operons. An operon is going to be a cluster of genes that are controlled by a single promoter. So that means there'll be one promoter and it will control gene expression of several genes that are all in a row. And this is typically due to environmental gene regulation. PROG shows you how to easily remember the parts of an operon, because an operon is going to be like a whole set of things that have to come together to control gene expression in these prokaryotic cells. The P in PROG stands for the promoter, R is the repressor, O is an operator, I'll talk about that in a second, and G is going to be the set of genes that are being controlled by the overall operon. These tend to occur in prokaryotes often. They are very rare in eukaryotes. So it's not that they don't occur in eukaryotes, just it's not very common. So most of the time we will be talking about operons in bacteria cells. It's just way more common. So it will involve a repressor protein. So that part is going to be really similar to what we just learned about on the last slides. But this time the sequence on DNA that the repressor protein is binding to is not called a silencer. This time it's called an operator. So when we look down here, we can see all the parts. Okay, so here's the genes right here. Genes 6, 7, and 8, those are the parts. So those 3 genes are going to be controlled by this single promoter, which is number 3. Number 1 is RNA polymerase. Number 4 is the part of the DNA molecule that's called the operator. Okay, it's part of the operon, but it is an operator. Try not to get those two mixed up. Bound to the operator in this picture is going to be the repressor protein. So you can see the repressor protein is in place. It is bound to the operator, and when this happens, RNA polymerase is blocked. So this operon is in the off position. This operon is the lac operon. The lac operon is a classic example of an operon in prokaryotes. Almost every textbook is going to talk about the lac operon and then maybe the trip operon. So we'll talk about both of those as examples of operons in bacteria cells. So again, the whole thing, right, the whole complex, PROG, is the operon. When we look down here, you can see that something's different, right? There's something present in the environment that is causing the repressor to not be bound to the operator. And so this operon is on. It's on because there's lactose present in the environment. The job of the lac operon is to create enzymes that break down lactose. Lactose is a disaccharide. It's a sugar that is made up of 2 smaller sugars. And so it's going to allow for, when lactose is present in the environment, it binds to the repressor, which leads to a shape change in the repressor. That causes the release of the repressor from the operator. Now RNA polymerase is no longer blocked. This means RNA polymerase will go down the gene, transcribing these 3 genes. Then they will be translated into enzymes that will break down lactose. Once all the lactose has been broken down in the environment, then the repressor will go back into position and turn the genes off, and it will go back to this particular image above right here. And so this is a way for bacteria to turn genes on and turn genes off due to what's happening in their actual environment. They would have no reason to make enzymes to break down lactose if there isn't any lactose in the environment. That'd be a waste of its resources. So this next video that you're going to watch is going to be a 10-minute video where Mr. Anderson not only talks about the lac operon, but he also talks about the trip operon. So you should take notes on your note taker about each of these things because they work a little bit differently. They do have all the same parts. All operons are going to have that same general structure, but how they work and why they're working is different. The lac operon is always in the off position and has to be turned on, but the trip operon will be the opposite. It's always in the on position and has to be turned off. Also take notes when Mr. Anderson talks about the regulatory gene. Take notes on that as well.
Hi, it's Mr. Anderson, and in this video I'm going to talk about the operon. The operon was discovered in the 1960s by three French scientists, Jacob, Rouff, and Monod. That's really bad French. But they got to coin the term operon, and basically it means to operate. And so it's mostly found in prokaryotic cells, mostly found in bacteria. There's a few eukaryotic operons, but in general it's just going to be found in bacteria. And so it led to other terms that you've maybe never heard of before, like regulons and modulons and stimulons. Stimulons are groups of operons operating together from one stimuli. And so it just means to operate. That's what an operon means. And so the one that they discovered was the lac operon. They were studying this in E. coli. And so the lac operon is designed for E. coli to break down lactose. So lactose is going to be a disaccharide. And remember, if you're a bacteria living in our gut, you're eating whatever we're eating. And so as the material moves down through our digestive system, they have to be ready to quickly shift depending on what the food is. If it's sugar, they have to be able to break down the sugar. If it's proteins, they have to be able to break it down into amino acids. And if it's a disaccharide, in this case, they have to move it into the cell and then break it down and chemically digest it. And so they use a lac operon to do that. Now, this is a picture of prog, but that's not how you spell prog. But it is the way that I remember the parts of an operon. You've got a promoter, a repressor, an operator, and then genes. And so if you can remember prog, it's going to get you half of the way there to remembering how an operon works. And so the neat thing about bacteria is that if they have a number of different genes that are used to achieve a certain task, they'll put them right next to each other. And so as the RNA polymerase moves down the DNA and makes all of the messenger RNA and makes all the proteins, you're just ready to do whatever the job is. And so the genes are going to be located right next to each other, in this case in the lac operon, to deal with lactose. What else do we have? Well, we've got the promoter. The promoter is going to be the region of the DNA where the RNA polymerase can grab onto the DNA. We also have an operator, and so think of that like a on-off switch that's going to turn on or off the operon. And then the last thing we have is a repressor. And so this is a lac operon. It's prog. It's promoter, repressor, operator, and then all of the genes. And so what are we trying to break down? We're trying to break down lactose or deal with lactose. Remember, lactose looks like this. It's a disaccharide. But in this little model right here, we're going to represent it as a little pentagon, a yellow little object that looks like that. And so all of a sudden, a lot of lactose is present. So what is the lac operon going to do? Well, it's going to deal with that lactose. And so all of this lactose is moving around, remember, but eventually one of the molecules is going to bump into the repressor. As it bumps into the repressor, it's going to change the shape of that repressor. And so a quick digression, everything promoter, operator, and genes are DNA. So they're going to be part of the DNA of the bacteria. But the repressor is a little bit different. It's a protein. It's a protein that came from a different part of the DNA. There's going to be a regulatory sequence, either upstream or downstream of the operon, that creates the repressor. It plugs in nicely to the operator, but once the lactose fits inside it, It's going to change the shape of that repressor. What does that do? Well, it opens up a region where that promoter can allow the RNA polymerase to get on, and then that RNA polymerase is just going to drive right down the DNA. It's going to make a bunch of RNA. It's going to make a bunch of those proteins. What are the proteins designed to deal with? Lactose. And so they're going to break down the lactose. Now all the lactose is gone. What's going to happen to that repressor? It's going to return to its original shape, and that shape is going to fit into the operator. And so if there's no lactose present, we would say the operator is in the off position because the repressor is activated. It's activated. It's sitting inside the operator. It's physically breaking the movement of that RNA polymerase. The RNA polymerase can't move down and code for those genes because the repressor is sitting in its way. What happens next? Well, lactose shows up again. Lactose would move into the repressor, free it up, and then it's going to be able to make those genes. And I've got a simulation at the end of this video that shows you in a little more detail how that takes place. Before we get there, I want to talk about the trip operon. The trip operon does essentially the opposite of that. So the trip operon is evolved in bacteria to deal with tryptophan, or more specifically to deal with the absence of tryptophan. So tryptophan is going to be an amino acid, and it's required to make proteins. It's one of those 20 essential amino acids. And so the trip operon basically is designed to make tryptophan if it's not present. And so we're going to get tryptophan in poultry, in milk for that matter. There's going to be high levels of tryptophan. But if a bacteria doesn't have tryptophan, there's a number of different genes that are required to make it, and so they can make their own. And so we've got an operon called the trip operon. How does that work? Well, again, we've got prog, we've got our promoter, our repressor, our operator, and then our genes. But if we have tryptophan present, so tryptophan is going to be these yellow hexagons, if we have tryptophan is going to fit inside the repressor, and it's going to change its shape so that it fits in the operator. And so in this case, if we have a bunch of tryptophan present, then we don't want to make tryptophan. And so the repressor is going to set that operator to the off position. What happens if all of a sudden the tryptophan goes away? If there's no tryptophan in the diet, well, the bacteria is not out of luck because it changes now the shape of that repressor. And so once it's changed the shape of the repressor, RNA polymerase can grab on, drive down that operon, make all of those genes, and those genes can be used to create more tryptophan.
With more than 55 degrees and certificates to choose from, 100% online classes, and personalized support.
And so it's a great kind of a feedback loop. So in a lacopteron, we're going to turn it on if we want to break down lactose. In a trip operon, what we're going to do is create tryptophan if we don't have it or if it's not present. And I really didn't understand how operons worked until I stopped thinking like a scientist and really started thinking like an engineer. We've got a problem that we have to solve, and one of the great ways we can do that is using an operon. And so I'm going to show you a quick simulation of how this works. It's a PHET simulation. Here's a website. I'll put a link to the simulation down in the video description down below. But it's a great way when I've used it with my students for them to wrap their head around how an operon works. And so we're looking at a lac operon. And to make it simple, we're only dealing with one gene, one of those three genes that are found. And so if we're to look at the simulation, you could try to figure out what everything is, but it's easier if I show you the legend and show you what everything is. And so we've got RNA polymerase kind of bouncing around here. Again, they'd be moving around. There's molecular motion. They'd just be randomly bouncing around. But this is one of the players. We've got the RNA polymerase right here. We could look down below, and I could ask you, where is the operon? Is this the operon here, or is this the operon? Well, the right answer is this. This is going to be the operon here because it's got a promoter. So let's put that promoter out here. It's got an operator, an on-off switch, and then it's going to have a gene, in this case the LAC-Z gene. So what's this up here? Well, this is not an operon, but this is going to be another what we call a regulatory sequence. And so this one down here is going to make an enzyme that's going to break down lactose. But what's this RNA polymerase up here going to make? Well, it's coding, it's making a little bit of messenger RNA, but what it's eventually going to make is it's going to make a repressor. Okay, so now we've got the fourth part of our operator. We've got the promoter right here, we've got the repressor, we've got the operator, and now we've got the gene. But you can see that the RNA polymerase can't make that gene because the repressor is blocking its way. And so it's in an off position at this point. And it should be. In other words, we shouldn't start making those genes until lactose is present. And so let's add a bunch of lactose. So I'm going to add a bunch of lactose here, and let's watch what happens. Well, if we add a bunch of lactose, and we could speed up the simulation a little bit, what's going to happen is the repressor, and I could pause it right here, that repressor is now having the lactose bind to it, and it's changing its shape, or it's changing its conformation. Once the lactose, and you can see it's happened in two cases, once the lactose binds to the repressor, it can't bind to the operator anymore. And so if we play the video, what's going to happen? Well, now RNA polymerase can drive down the operator, down the operon. It can produce a protein. In this case, that's going to be a LAC-Z protein. What's it going to do? It's going to digest and break down all of the lactose that's present. And you can see that it's gobbling up all of those lactose. The only lactose left is those that are bound to the repressor itself. What happens eventually, all of the lactose is going to disappear. In other words, we will have broken it down. What happens now? Well, that repressor is going to bind to the operator again, and now we can't make that protein to break it down. And so it's a wonderful feedback loop. It's a feedback loop that deals with a bunch of lactose present. Because E. coli would be silly to make all of the proteins to break it down until we've got a bunch of lactose present. And so that's an operon. What is it again? It's all of the genes and then a way to control those genes in one tidy little package. And I hope that was helpful.
Okay, so the lac operon, just as a recap, is a inducible operon. That means it's naturally going to be in the off position and it has to be turned on. It gets turned on by the presence of an inducer, which would be lactose in that case. That way it can break down the lactose in the environment. And the trip operon is opposite. It's considered to be a repressible operon, which means it's always in the on position, normally making tryptophan. But once there's enough tryptophan present in the environment and it does not need to make any more, tryptophan binds to the repressor protein, which then binds to the operator, turning it off. Remember, he did talk about the regulatory gene. I would make sure you have written that down in your note taker. The regulatory genes are not the genes that are encoded for by the operon. Those are the genes or the structural genes, something like that. But the regulatory gene is outside of the actual operon, and what it codes for is the repressor protein. So the regulatory gene codes for the repressor protein that will be used to turn the operon on or off. Next topic is how environment can regulate gene expression. These can be physical or chemical factors present in the environment. We already talked about the lac and trip operons as examples of this. This is an example of environmental regulation because the entire operon is controlled by the presence of lactose or tryptophan in the environment. An example of something physical that can regulate gene expression is the sun can interact with your skin cells and cause melanin production to be turned on. Melanin is the protein which puts pigment into your skin cells. And so the more exposure to sunlight, that increases the transcription and translation of the melanin protein, and so you will get a tan during this time. Less exposure would decrease that process. Hormones are good examples of environmental regulators because they typically bind to receptors in the cell membrane, and then that leads to signal transduction pathways inside the cell to trigger a change, typically in gene expression. And so we've talked about this in the plants unit for sure, using auxins. Auxins play a very important role in the formation of leaves or the formation of flowers in the apical meristem. And these changes to go from leaf to flower are due to changes in gene expression that would be triggered by auxin hormones. In IB Year 2, you will talk about other types of hormones, such as testosterone, and how they are used to regulate gene expression. There are all kinds of molecules present in the air due to air pollutants that can cause an effect on the methylation patterns inside of the cell, and therefore can affect what's called the epigenome. And so this is going to be a really good example of how environment can interact with your DNA, causing changes in gene expression. And we will talk more about this on upcoming slides. Epigenetics is going to be part of that phenomena where environmental factors can interact with DNA and change gene expression. So first we'll talk about epigenesis. Epigenesis is a process that multicellular organisms go through that involves changes in gene expression throughout in a cell's lifespan or cell division processes, which ultimately are going to cause those cells to become specialized cells. Remember that you start off as a single-celled zygote, which is completely unspecialized. You go through several rounds of cell division, and you're still just stem cells that are not differentiated at this point. At some point during your development, though, even as an early embryo, we start to see changes that were not present, structures that were not present in earlier stages of that organism's life cycle that continue to change and develop over time. This is because your cells are now becoming differentiated or specialized, and therefore they're going to have specialized functions, and this is due to changes in gene expression. So another really good example of these developmental milestones that we can see is when a frog is going through its life cycle stages, where it's a tadpole, and then it has legs, then it loses its tail, now it's an adult frog. All of that is due to epigenesis. And these genes are going to be turned on or off by epigenetic tags. Epigenetic tags are going to be methyl groups typically, or other types of chemical groups that are going to be attached either to the DNA or to the histone proteins that associate with DNA in eukaryotic organisms. These epigenetic tags don't change the genotype of the organism, they only affect the phenotype. So no change to the actual genetic code, but a change in how that code is being expressed. Epigenetic tags can be attached to nucleosomes or directly to the DNA. Remember, a nucleosome is going to be a bundle of histone proteins. There are 8 histone proteins. You can only see 4 here, but there are 4 more behind it to create this spherical structure. And then your double helix wraps around it 2 times. So something we know about eukaryotic DNA is it's always associated with these histone proteins. And this is not true in prokaryotic DNA. So this is a nucleosome, here's another nucleosome, here's another nucleosome. In BioH, we talked about the importance of these nucleosomes in the formation of a chromosome. So when your DNA goes from loose chromatin to tightly compacted chromosomes during cell division, nucleosomes are going to help with that. But nucleosomes have other functions too. They help in gene regulation. So genes can be turned on or off depending on the type of epigenetic tags that have been attached or unattached to the histone proteins. So we could be talking about acetyl groups or methyl groups, or there's other types of tags too, like phosphate groups and so on, but we will focus on these two. When acetyl groups are attached, those are shown in blue, it causes the nucleosomes to be more spread out, so they're more spaced apart, and this causes the gene itself to be more accessible to RNA polymerase, which is the enzyme that needs to get in there and transcribe the gene. So when the nucleosomes are spread out because they've been acetylated, the gene is on. RNA polymerase will transcribe it, and therefore it will be translated. Methyl groups can do that. They can spread them out like this and turn the gene on, or depending on the gene, they can also cause the nucleosomes to be all scrunched up together, which would cause less access to the gene, turning the gene off. So methylation can do either if it's attached to a histone protein of a nucleosome. Methylation can also occur to the DNA itself It would typically occur to the cytosine nucleotide base pairs, but it can also occur to adenine bases as well. It usually occurs to bases that are close to the promoter, since that's where polymerase binds to, and this time methylation will always decrease transcription if it's attached directly to the DNA. There's no pictures on this slide showing that type of attachment, but there will be one on the next slide that I'll point out to you. These are typically reversible, which means cells have capabilities of adding these methyl groups to the DNA and then also removing them using enzymes. Epigenetics is like the general study of you inheriting these tags and passing them on to your offspring. Remember that these tags don't change the actual genetics, they don't change the genome itself, but they do change the way your genes are expressed. So we do call this the epigenome. The epigenome is going to be all the epigenetic tags in a cell or organism, and that plays a role in gene expression. This picture shows you how those tags can be attached to the histone proteins, like in this picture, or directly to the DNA if it's a methyl group, like in this picture. Again, nucleosomes can have the different groups attached to them. These are the epigenetic tags, and when the tags open up the nucleosomes to make them more spread out, then that turns the gene on. But when the tags that are added cause the nucleosomes to scrunch up together, that would impact transcription and turn it off. Epigenetic imprinting is a concept that does involve epigenetic tags, but involves the inheritance of epigenetic tags from only one of your parents. So you have all kinds of tags that you gather during your lifetime because of environmental conditions and so on. But during the formation of sperm and egg, most of these tags will be completely erased from your DNA. This is called reprogramming. So during the reprogramming phase, about 99% during meiosis of your tags are stripped away, and only 1% of your tags remain on. These tags result in imprinted genes. The tags that stay in place in sperm versus the tags that stay in place in egg are different. So you will inherit some imprinted genes from your mom and some imprinted genes from your dad. Most genes we inherit as two working copies, which means neither of the genes are imprinted. These genes are called biallelic, bi meaning two, allele meaning the allele, the version of the gene. So we've been talking about this always. In BioH, we said you could be big A, you could be big A, little a, you could be little a, little a. Those are the two alleles, and they're both on. They're both working, making proteins. Those are biallelic genes. Most of your genes work this way, but some of your genes are going to work in an imprinted fashion, where one gene is imprinted with a methyl group typically, and the other gene remains active. Methylation in this case is going to repress the gene, turn it off, silence the copy. And so these are called monoallelic genes, meaning one allele is on and one allele is off. The allele that's off is the imprinted one. Some of these you inherit from mom, and some of these you inherit from dad. So the silencing, the epigenetic silencing using the methyl groups, remember, is happening during the reprogramming phase of meiosis, during sperm or egg formation. Then sperm and egg come together for fertilization, and we get that zygote. So that zygote now has tags in place on the imprinted genes, some from mom, some from dad. Those tags are meant to be imprinted for the rest of your life. So those imprints are going to stay on for the entire life of the organism. You will also acquire other tags during your lifetime, and those tags get stripped away and they get reset once again during the formation of sperm and egg. So this shows tags, the imprints. Remember, you have all kinds of imprints or tags, not just imprints, but all kinds of tags on your DNA that you acquire throughout your lifetime. But in the reprogramming phase, most of them get stripped away and only some remain in place that would get passed on to the offspring. Those are the imprinted genes, and they are going to be different in sperm compared to egg. So here shows, we're making sperm and egg. You pass on your genes to your offspring. Here's fertilization. And this gene up here, the A gene, it's biallelic. Both genes are on. It's a non-imprinted gene. But in gene B down here, there is an imprinted copy. You inherited that imprinted copy from the sperm. The egg's copy is on. So this is a monoallelic or imprinted gene. Some genes are meant to be inherited that way. You have about 25,000 different genes, but only 156 of them are imprinted. You don't need to know these numbers, but remember, you do inherit specific imprints from the maternal chromosomes and then different specific imprints from the paternal chromosomes. Here shows a biallelic gene. They're both expressed, and these are both monoallelic genes. This one, mom's copy is on, and dad's has been epigenetically silenced, and this one is the opposite. These gene pairs must be, in fact, inherited in this monoallelic fashion. Without inheriting the imprinted genes, the embryo will probably not develop normally. So they are important. This example is going to go through a mutation on a chromosome, specifically in chromosome 15. In this region of the chromosome, this section shown here, there happens to be 2 imprinted genes that are right next to each other. So this doesn't happen often, but on this chromosome, that's where the imprinted genes are located. You are meant to inherit an imprinted U gene from your dad, and then the U gene remains on from your mom. You are meant to inherit the imprinted S gene from your mom, and the S gene is on from your dad. So again, they're right next to each other in this region of chromosome 15. This is normal. If a mutation occurs that results in a deletion of that section of the chromosome, then it would matter which chromosome had the deletion. It would matter, like, did you inherit that mutation from the sperm or from the egg? And it would result in a completely different disorder if you inherited it from one parent versus the other. So in this example, the mutation has been inherited from the paternal chromosome. So they have an active U gene, but they don't make the S protein at all because mom's has been epigenetically silenced. The result is called Prader-Willi syndrome. These individuals tend to have weak muscle development, and they also tend to develop childhood obesity starting at a very early age due to an uncontrollable, insatiable appetite. If you inherit the same exact mutation, but from the maternal chromosome, now you don't actually make the U gene, but this time you do make the S protein. So because of that, you have a different disorder because you're missing a different protein. This disorder is called Angelman syndrome. It's a nervous system disorder. It's characterized by a lot of laughter. They're often called like the happy children. They always smile, they always laugh because their nervous system is not operating correctly. But it also develops or leads to some form of mental retardation and delayed development, and then many seizures. This is a specific IB example, so you should make sure that you do know this example, maybe write IB. There are imprinted genes present in other types of mammals like lions. And so we're going to talk about the effects of that. So in nature, male lions are passing on genes that encourage growth, right? That say you will be a large lion. This is because they want to maximize the fitness of their offspring, and they're always competing with other male lions to mate. Female lions, though, mate with multiple males, and in order to ensure that all of her offspring survive, she actually has imprinted genes that she is passing on that kind of suppress that growth gene from the male. So it's like an anti-growth gene, and this is an imprinted gene that she passes on to all her offspring. This allows all her cubs to potentially grow at the same rate, and hopefully all of them will be born healthy. In tigers, they don't have the same issue because a male tiger and a female tiger just mate with one male, or just like one mating pair at a time. So the male tiger is not in direct competition with other male tigers, and so he's not passing on, these heavy growth genes because he doesn't need to. And the female tiger, therefore, does not have to have competition in the same way that a lion would, so she does not have any imprinted genes. So she doesn't have the anti-growth gene either. In nature, lions and tigers do not typically mate with each other. However, in captivity, they sometimes do, and they do have viable offspring. If it's a male lion mating with a female tiger, we call their offspring a liger. So the hybrid is a liger, and these hybrid offspring are usually huge, much bigger than even the male lion parent. This is due to two things. One, the male lion is passing on genes that encourage growth, and two, the female tiger is not passing on any imprints that discourage that. Therefore, their offspring tend to be very large in body size. The tigon is when a male tiger and a female lion mate with each other, and their offspring tend to be smaller in size, sometimes smaller than the parents or at least the same size. And this also is due to two reasons. One, the male tiger is not passing on growth encouraging genes, and the female lion is passing on an imprinted gene that is suppressing growth. Therefore, growth is not encouraged. It's actually, they tend to have smaller offspring. And so this just shows that male lion, female tiger, no imprints, growth genes being passed on. The liger is huge. Male tiger, female lion, just normal growth genes being passed on, but the female's passing on the imprinted gene saying be small. And so the tigons tend to be small. This is just another picture, again, showing the difference in body size between ligers and tigons due to imprints that are either passed on or not. Nature versus nurture is probably something you've heard of before. This debate is in reference to how much of your traits are expressed due to your actual genetics, like your DNA. So that is going to be the nature part. And then nurture is your environment. How much does your environment play a role in how your traits are expressed? And so scientists like to study monozygotic and dizygotic twins. They really like to study monozygotic twins. Monozygotic twins are known as identical twins. These are the types of twins where they came from one fertilized egg that split into two and then developed into two organisms. So they are genetically identical. They have the exact same DNA. Dizygotic twins are known as fraternal twins. They came from two eggs fertilized by two different sperm. So they are as genetically different as two siblings that have different birth dates, for example. They're simply siblings. So again, scientists like to study twins because they can study twins that were raised together that would then have the same genetics and likely have the same or at least similar environments, but also twins that were raised separately because they would still have the same genetics but could potentially have very different environments. And so then they can study how much does environment actually play a role in specific disorders. Identical twins will have the same genome, but they will have a different epigenome. But by how much does that epigenome vary? We would expect for it to vary a lot more with twins that are raised separately. This is showing concordance rates between twins. Identical twins are shown in red, fraternal twins are shown in blue. Concordance rates means if one twin has it, the other twin also has it. So you can see that there's a genetic component to some more so than others. And then you can also see that something is affecting the organisms that's beyond genetics because, for example, in this example, alcoholism in females, 30% of female twins, one twin has it, but that means in 70, and the other twin also has it, but in 70%, one twin has it and the other one doesn't. So what's causing that difference in identical twins? And so usually they attribute it to environment, and there's a lot of disorders specifically that they're studying to see how much of a role does environment play in those specific conditions. So things from autisms to depressions to other disabilities, to even cancers. Something else that helps to control gene expression is the regulation of how much mRNA is inside of the cell. So mRNA can stay in the cell for varying amounts of times depending on the needs of that cell. Could be minutes, could be days. Kind of depends on how much protein does that cell need of that particular type of protein. There are going to be nucleases present in the cytosol that can break down or degrade the messenger RNA when it's no longer needed. These nucleases start at the poly A tail and they start breaking off the poly A tail. The shorter the poly A tail gets, the less likely that messenger RNA is to be transcribed. Once they start cutting into the nucleotide sequence, it will no longer be transcribed. And so this is a way to control gene expression by getting rid of the mRNA when they're no longer needed. This also helps to recycle the nucleotides so they can be reused for the cell. The last process that regulates gene expression that we'll learn about is called alternative splicing. Alternative splicing means that, remember we talked about regular splicing, which is where we remove the introns and splice the exons back together. But if we only use some of the exons, then we can actually make different proteins from the same gene. So this increases the amount of polypeptides that can be made from a single gene. This is one of the reasons why you have about 25,000 genes, but your body codes for over 200,000 different types of proteins, alternative splicing mechanisms. So the example here is, there are 5 forms of tropomyosin, which is important in muscle cells. And this is going to be based on these different types of muscle fibers are going to be based on different types of splicing. So if we use only some of the exons, we would get smooth muscle. If we use different exons, we could get skeletal muscle, and so on. This one shows the same thing, alternative splicing, right? We're making protein A, protein B, and protein C. There are 5 different exons here, so you can see that this one we're using all 5, this one we've skipped 3, this one we've skipped number 4, and so on. And so we can get different proteins because of that. Alternative splicing, splicing the mRNA in different ways or alternative ways to increase the number of different types of proteins that are being created from a single gene. And then this is going to be an animation to help support alternative splicing.
Hi everyone, and welcome to Biology Professor. Today's lesson is about alternative splicing. Now, first let's talk about what just regular splicing is. Splicing is a post-transcriptional process in which introns are removed and exons are joined together. What does this mean? This means that in a pre-mRNA transcript that's made in the process of transcription in a eukaryotic cell, there are introns, which are non-coding regions, exons, which are coding regions. The introns get spliced out. This can confuse students because in versus out, but in stands for intervening sequence, X and the exon stands for expressed sequence. So it's the exons that are joined together into the mature transcript. Now, alternative splicing is a regulated process where there are multiple protein isoforms that can come from the same pre-mRNA transcript. So these multiple protein isoforms are encoded by a single gene via exons, parts or exons, or introns being differentially joined or skipped. Now let's look at a picture of this. Right here, we have an mRNA transcript. It needs to be spliced. The exons are the parts that have different colored patterns, so and the white portions are the introns. So the introns get spliced out. Some of them are degraded. Some of them have regulatory processes, which we'll touch on in just a minute. But the exons, you see there's 4 exons here, and in this example, they can be differentially joined or skipped. Differential, this is one of the reasons why a synonym of alternative splicing is differential splicing, but they can be joined or skipped in different ways. So for example, in this isoform, we have this exon, and this exon, but this one has been skipped. In this isoform, we have this exon, and this exon, but this one has been skipped. So in alternative splicing, you have situations where multiple protein isoforms, like 2 shown here, and often even more than 2, can be encoded by a single gene. And this occurs because exons, or even different parts of exons, or introns can be joined or skipped in different ways, resulting in a lot of different combinations. So this isoform and this isoform, remember that these are still mRNA transcripts. They have not been translated into protein yet, but they do code for different proteins now, because this mRNA transcript and this mRNA transcript have different sequences. The fact that they have different sequences means that once they are translated at the ribosome into protein, those proteins have different structures. Now the structures can be related. For example, they both have this component here. They both have this component here, although in this one they're separated, in this one they're together. This one has a different component at the front. So they've got different structures, related structures, but certainly with differences. And these different structures can lead to different functions. And so you can have these different protein isoforms that are all from the same gene, but because of alternative splicing, they can carry out different functions in the cell. And I also want to take a minute to talk about the fact that in humans, as many as 95% of multi-exonic genes, that means genes that have more than one exon in the transcript, so 95% of multi-exonic genes are alternatively spliced. So this process is a very, very common one in most eukaryotes, including humans. And the really great thing about alternative splicing is that it very, very much increases the diversity of proteins encoded by the genome, because you can get proteins that, while they might have related structures, they have different functions, all coming from the same gene. And this is why when the human genome was first sequenced by the Human Genome Project, scientists were very, very surprised, shocked to find that such a small portion of the human genome actually codes for proteins, and so much of the rest of the genome is regulatory DNA. Some people will call it junk DNA, but we know now that that's not really true. And people thought, well, if such a small portion of the human genome codes for proteins, how do we get so many proteins? And then we learned about alternative splicing, and suddenly it all made sense. Let's talk just very briefly about the mechanism of alternative splicing. It is highly, highly regulated. It's not something that just sort of goes off randomly. It is regulated in part by sequences within the mRNA transcript. I said earlier that when these introns get spliced out, sometimes they're degraded, but other times the introns themselves, which are just a sequence of nucleotides, can be highly regulatory. They can inform other components acting in this mechanism on how to act, where to cut, where to join. Also, there are various proteins that play a role, specifically 2 types of proteins. The first are activator proteins. The second are repressor proteins. And these do what you might think. The activator proteins activate certain aspects. The repressor proteins repress certain aspects, and this controls which places get cut, which places get skipped, which places get joined. And also a major component of this process is a large complex of RNA and protein that work together. This complex is called the spliceosome. And the spliceosome works along with the activator proteins which activate it, repressor proteins which repress it, to go and cut in the right places and join in the right places to get these different transcripts that eventually lead to the different protein isoforms. So that is it for this video on alternative splicing. If you're interested in learning about post-transcriptional modifications like regular splicing, also 5' capping, the 3' poly A tail, please see my video on the post-transcriptional.
So that is the end of the gene regulation notes. You don't need to know all the tiny details she discussed on the alternative splicing video, but I think that will kind of help sort of bring it all together for you.